Hey folks, okay, I'm going to lead you through what you need to do to catch up if you missed the day that we started the Potato Diffusion Lab. And actually the first thing that we did that day was we did a little bit of work with the reference tables. So what you need to do is find page 8 in your reference tables that I gave you about a month ago. Page 8 starts with table T, and we're interested in table U, V, and W. So hit pause and go find page 8 in your reference tables. Okay, now that you've got page 8, we're going to mark it up a little bit, and you can use this marked up version for any tests and exams this year, except for the final exam. So some of these charts have some things that are confusing, but some things that should be kind of familiar. Like you've heard of the word enzyme. Well, these are some different enzymes here. And this column here says acts on. I want you to write in breaks down. What that means is amylase is an enzyme that breaks down starches or breaks them apart. Lactase is an enzyme that breaks apart complex sugars. Pepsin is an enzyme that breaks apart proteins. And this column just tells you where that happens. So everything that I put in red, you write down on your chart. Then if we jump down here, remind yourself the word organic means it's something that contains carbon. And compounds, those are where you have two or more elements bonded together to make a substance called a compound. Now, in this chart down here about energy-rich organic compounds, the word macromolecule, that just means a big molecule. Proteins, carbs, lipids, and nucleic acids are really, really big molecules, okay, made of thousands of atoms. Subunits just means the smaller molecules that these big molecules are made of. Like a protein is made of lots of amino acids attached together. A lipid is made of lots of fatty acids attached together, okay? Now, I want you to jot down in table V that remind yourself that a starch is a complex carbohydrate. Simple sugars are things like glucose, okay? They're simple carbohydrates or simple sugars. Okay, now that you have marked up your reference table, you're going to take the white sheet that I gave you called Nerding Out with Nutrients in the Reference Tables, and you're going to use that marked up reference table to work through these problems and questions. Start with questions 1 through 5, and those should all come from table U, okay? And then you're going to work your way through questions 6, 7, 8, and 9, 10 and 11, 12, skip 13, and once you've done that, you're going to go to the instruction sheet, find the answer key, and you're going to check your answers, okay? And so hit pause while you go and do that. All right. Now you're back. Okay, I'm going to lead you through the first part of the Potato Diffusion Lab. It's a blue packet, so find that blue packet that I gave you, okay? Now what I'd like you to do first is take a few moments and read the top half of the front page, the very top half, where it says Introduction, okay? And hit pause. All right, now you're back. Well, you should know now that the job of the cell membrane is to protect the cell. It's also to let in good molecules and let out bad molecules or waste molecules. Because a cell membrane, really, it isn't totally solid. It has little holes in it. It really is kind of more like a net, and it can let things through that are small enough. Now, when things travel through a membrane, that's called diffusion, and you should see a big vocab reminder about the word diffusion. Diffusion means the movement of molecules across a membrane, such as into or out of a cell. There's a big vocab reminder on your blue packet on the front page. All right, so today we're going to work with cells that are in a potato. A potato, if it's not cooked yet, is made of lots of cells that are still alive, lots of plant cells, okay? And if we zoom in, they would look something like this. They're kind of boxy because they are plant cells, and they have a cell wall and a cell membrane. All right, now, diffusion, we said it means molecules moving across a membrane. Now, there's sort of a rule of diffusion that you need to know. And right here in our diagram, this would be a membrane. Here are some molecules on one side of the membrane. Here are some molecules on the other side. Now, molecules prefer to move from areas of high concentration to areas of low concentration. They like to even out, okay? Now, over here, this is an area of high concentration with lots of molecules in that amount of space. Low concentration means you have fewer molecules in that amount of space, okay? And I'm going to show you what would naturally happen. Molecules like to even out. So some of these molecules would just move right through the membrane until they're basically evened out on both sides. Okay, so see now they're kind of even on both sides. We have, 8 molecules on both sides. And they can keep moving back and forth, but now at this point, they like to stay evened out. Okay. All right, now a couple of terms that we need to bring in, maybe from gym class. Which of these people is being active and which is being passive? Well, you could probably figure out that the person over here who's using energy, they're being active. This person who's not using energy, they're being passive. And we want to associate those terms. Active means using energy, passive means not using energy, because those terms are important in diffusion also. There are really two kinds of diffusion. You can have active transport, and that's where energy is needed to move molecules backwards from low concentration to high. So if a cell wants to move molecules against the way that the molecules would normally go by diffusion, the cell can use energy to do that. But passive transport, it means no energy is needed. The molecules just diffuse from high to low concentration the way that they naturally would. Passive transport is basically plain old diffusion. Active transport is when a cell uses energy to make it go backwards. Okay. So what your classmates did in this lab is we took 2 chunks of potato and we measured the mass of each chunk, and then we recorded the mass on our lab sheet. Now, in a minute, I'm going to give you some data so you can play along. Then we put each chunk of potato in a different solution. One was in fresh water, 100% pure water. The other was put in salt water, which was about 50% water, 50% salt. And we left them overnight to see what would happen. Okay. And so in a moment, you are going to get some data so you can play along with what happened. Okay.